Good morning, student. We will continue the topic of hemoglobin, and today's topic is role of 2,3-bisphosphoglycerate in oxygen binding and delivery. First, we will see how this 2,3-BPG is formed. It is also called as Rappaport-Lubring cycle. Definition of this Rappaport-Lubring cycle is it is the diversion of one step of glycolysis. Site of this Rappaport-Lubring cycle is it occurs in RBCs. Here you will see a glycolytic pathway I have mentioned in short. In glycolysis, 1,3-bisphosphoglycerate is an intermediate which is converted into 3-phosphoglycerate. But in Rappaport-Lubring cycle in RBCs, this 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate with the help of enzyme Bisphosphoglycerate mutase. Here the phosphate group which is attached to the first position is first carbon is shifted to the second carbon. Then this 2,3-bisphosphoglycerate is converted into 3-phosphoglycerate by removal of phosphate group from second carbon in the form of inorganic phosphate. The enzyme required for this step is 2,3-bisphosphoglycerate phosphatase. Now this 3-phosphoglycerate then join the glycolytic pathway. So this Rappaport-Lubring cycle is one step diversion of glycolytic pathway in RBCs, due to which this 2,3-bisphosphoglycerate is formed inside the RBCs. One more thing about this Rappaport-Lubring cycle, if you see glycolytic pathway, when 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate, 1 ATP is synthesized. But here when Rappaport-Lubring cycle occurs, 1,3-bisphosphoglycerate is converted into 2,3-bisphosphoglycerate, and this 2,3-bisphosphoglycerate is converted to 3-phosphoglycerate. Here ATP synthesis does not occurs. Now what is the role of 2,3-BPG? This 2,3-BPG binds with 143rd histidine and 82 lysine residue in beta chain. Beta chain is a protein in hemoglobin. It is made up of various amino acids. At 143rd position there is a histidine amino acid, and at 82 position again one more amino acid is lysine. 2,3-BPG binds with these two amino acids, histidine and lysine. Now after binding with this amino acid, this 2,3-BPG decreases the affinity between hemoglobin and oxygen. Therefore hemoglobin when reaches the tissue, it can easily gives the oxygen to the tissue. So 2,3-BPG plays an important role in delivery of oxygen to the tissues. Now this 2,3-BPG has less half-life. Just we have seen it is readily converted into 3-bisphosphoglycerate. So it is constantly synthesized and metabolized in RBCs by glycolytic pathway. In this shunt pathway, no ATP is generated. Just now I have explained. So it prevents the accumulation of ATP, because RBCs does not require much ATP because it does not have any cell organelles. So prevention of ATP accumulation is seen due to this Rappaport-Lubring cycle. Role of 2,3-BPG in fetal hemoglobin. Basically, fetal hemoglobin 2,3-BPG have no role, because fetal hemoglobin or hemoglobin F is made up of 2 alpha chain and 2 gamma chains. Just now we have seen that 2,3-BPG binds only with the beta chain. So due to absence of beta chain in hemoglobin F, 2,3-BPG does not binds with hemoglobin F. Hence hemoglobin F has high affinity for oxygen. Just now we have seen 2,3-BPG decreases the affinity between hemoglobin and oxygen. As there is no binding of 2,3-BPG in hemoglobin F, affinity between hemoglobin F and oxygen is high. Hence hemoglobin F delivers less oxygen to the tissue, which is not beneficial for our body. This can be demonstrated by this example. When partial pressure of carbon dioxide is 27 mm of Hg, at that time hemoglobin saturation of HbF is 50%, whereas hemoglobin A shows 50% saturation with oxygen at PCO2 19 mm of Hg. It delivers less oxygen to the tissue, just I have told you, at PO2 40 mm of Hg, which is seen in the tissues. Therefore, there is shift of oxygen dissociation curve to the left in fetus. Role of 2,3-BPG in A2. Again, A2, hemoglobin A2 is made up of 2 alpha and 2 delta chains. It also does not possess beta chain, hence 2,3-BPG does not binds with hemoglobin A2. Hence, hemoglobin A2 also have high affinity for oxygen. It also delivers less oxygen to the tissues at PO2 40 millimeter of mercury, which is seen in the tissue, and there is shift of oxygen dissociation curve to the left. So this is the role of 2,3-BPG in hemoglobin A, hemoglobin F and hemoglobin A2. Thank you very much.